Do you know that bacteria can actually talk to each other and coordinate their group behavior? This phenomenon is called quorum sensing. Through quorum sensing, bacteria use the chemical signal molecules or autoinducers as their language to communicate. These autoinducers are released by the bacteria and accumulated in proportion to the number of bacterial cells. And once the bacteria population reaches a certain concentration or a quorum, the expression of associated genes will be activated. Lux system that was first found in Vibrio fischeri is often used to describe quorum sensing. In this system, Lux-I is the synthesized molecule that produces the autoinducers called N-acylated homoserine lactones or AHLs. In low cell density, the AHL will freely diffuse across the membranes and out of the cell. Independent Lux-R, which is the AHL binding protein, on the other hand, will be degraded inside the cell. During high cell density, the concentration of AHL molecules will be accumulated. When a critical threshold of AHL concentration is achieved, some AHL molecules can re-enter the cell and bind to Lux-R. The stable Lux-R AHL complex then binds to the Lux-R box promoter and initiates the expression of the target genes. In Vibrio fischeri, Lux-R system activates the expression of genes that encode for luciferase proteins. This causes the production of bioluminescence. In other species, AHL molecules can have slight variations that can only be detected by their own species and target different genes to be activated. For example, genes for biofilm formations and also virulent factor expressions. In aquaculture, quorum sensing is known to regulate virulence mechanism of many pathogenic bacteria in the aquatic environment. Therefore, many types of research have been conducted towards the development of antivirulence therapy to disrupt the quorum sensing components. The exploitation of bacterial quorum sensing is also hoped to be one of the solutions in tackling the antimicrobial resistance issues in this sector.